Hi everyone, welcome to the second part of our Chapter 3 discussion. As a general reminder, this chapter is entitled Cells, the Living Units. In our first part, the plasma membrane, we talked about the fact that it's composed mostly of phospholipids. It is in the form of a bilayer, and it can be described as a fluid mosaic model, and as well as its selective permeability capacities. What I would like to do in our final few slides on the cell membrane is talk about something called RMP. This is a resting membrane potential, and it's basically a voltage charge that will occur across the plasma membrane due to the movement in and out of different charged ions, primarily sodium, potassium. This is important to understand because it will lay the basis of generating an action potential, which is when the cell gets so excited that it's able to respond to various stimuli. Now, in order to understand the resting membrane potential, we have to remind ourselves that across the membrane, we have a continuous movement of sodium and potassium due to the sodium-potassium pump. And as these sodium and potassium ions are moving in and out, what they'll do is they'll bring along a charge with them. Depending on the concentration that is stipulated, what we see happening is that it can create a resting membrane potential, which is often indicated by a negative millivoltage number. And what the negative indicates is that the inside of the cell is more negative compared to the outside of the cell. Here in this particular illustration, we can see how we can explain the formation of this particular electrical potential energy. If you start on the left-hand side, you're going to notice that you have what are labeled as potassium leakage channels. And these are going to allow the charged potassium ions to diffuse down their concentration gradient and move out of the cell. As they're moving out of the cell, what we see happening is that we start to occur more of a positive charge happening as they are reacting to the formation of these new potassium ions. As the potassium is leaking out on the left, you're going to notice that on the right, there is a protein channel that has been drawn for you that shows that the potassium ions are moving back into the cell. And this is partly due to the fact that they are attracted to the charge that's been generated by their initial departure on the left side of the image. As these potassium as well as the sodium ions move in and out throughout, they then start to generate a resting membrane potential. This resting membrane potential can then be used to respond to a stimulus to generate an action potential, which is the response of a cell based on reaching the threshold stimulus that is required. Now, action potentials are an excellent way for us, for instance, to explain the muscle contraction, the beating of the heart, the response of a neuron, and these are going to allow us to explain how a cell can get excited to the point where it has a response to the stimulus, excuse me. In order to fully understand an action potential, what we see happening is that we are going to have to explain depolarization and repolarization. And these are going to indicate changes to the membrane voltage depending on which ion is coming in or out of the cell. So let's go ahead and take a look at a general image of an action potential. And if we look at our graph, we notice that we have the time, and of course we have the voltage that we are looking at across the cell membrane. Now this cell membrane has set a resting membrane potential of negative 70 millivolts. And as we can see across the time, if there's a stimulus that's received that can reach the threshold, we see that the voltage shoots up, aka the cell becomes more positive. Now, when the cell becomes more positive, this is due to the fact that the stimulus has activated sodium channels, and sodium is now coming into the cell. And as sodium travels into the cell, it allows the cell to gain a more positive voltage. Eventually, this is then going to be referred to as depolarization. So depolarization is when the millivolts go up, and that is because sodium is coming into the cell. That's depolarization. Eventually, the cell will become so positive that it will allow for an action potential to occur. So this is then the response that's happening from the nerve cells and from the muscle cells. Once the action potential is completed, what we see happening is that the sodium channels will then shut down, and instead potassium channels will get activated, and potassium will come out of the cell. And as it goes out of the cell, it allows for the membrane voltage to go down, and this is then called repolarization. Now, potassium floods out at a much quicker rate. So what we see happening is that repolarization, or the negative charge or the lowering of the membrane voltage, is going to happen at such a quick rate that oftentimes it goes a little bit more negative than its resting membrane potential. And this is now what we call a hyperpolarization. No need to worry, because what's going to happen is as soon as the potassium channels are closed, sodium will start back up again, and it will not only return it back to the resting membrane potential, but it will also ready the cell for another depolarization, which can lead to another action potential. So let's kind of review this concept. Just one second. Let me get rid of all this. In order for your cell to generate an action potential, it needs to have a resting membrane potential, which can be stimulated. And when the resting membrane potential is stimulated, that means that the voltage becomes more positive, so it shoots up. This is called depolarization, and depolarization is due to the fact that the sodium ions will enter into the cell, thereby exciting the cell, becoming more positive, going through depolarization, which will eventually lead you to an action potential. Once the action potential or the excitement is done, then the cell has to reset itself. So we're going to be doing repolarization, which means we're going to go back down to our resting membrane voltage. And in order to do that, we need to find potassium channels, open them up so that the potassium can come flooding out of the cell. And since the potassiums move at a much faster rate, oftentimes the repolarization step will cause the cell membrane to become slightly more negative than it was before. This is called hyperpolarization. And as I mentioned before, nothing to worry about, because as soon as the potassium is closed and the sodium switches on, it will go ahead and restore it back to the resting membrane potential, as well as ready the cell for another depolarization process. Please take a look at this chart and please understand what the action potential is, and that it entails the movement of the sodium into the cell membrane, getting it all excited, depolarization, and then once you've reached your action potential, you will then need to reset or repolarize, which means that potassium has to come out of the cell. Beautiful. So having said that, let's go ahead and let's just kind of shift our conversation more to our cytoplasmic organelles. So we know now about the plasma membrane. Let's focus on what's happening in the cytoplasm, that area between the nucleus and the plasma membrane in a eukaryotic cell. I had mentioned in part 1 that you will find lots and lots of cytosol there. This is the gel-like substance in which the organelles are embedded. And then what we're going to do now is we're going to take a look at how these organelles function, as well as mention some what they call inclusions. Inclusions are structures that do not have a separated membrane. So these can be things like pigments, crystals, vacuoles, or debris that will be floating around. For our organelles, we are going to mostly focus on the mitochondria, the ER, or endoplasmic reticulum, the Golgi apparatus, and we're going to talk about peroxisomes and lysosomes. And then for the ones that don't necessarily have a membrane, we're going to take a look at the ribosomes, the cytoskeletal, and the centrioles. For our exam, for those of you who are listening to me from our A&P course at FAU, you want to make sure that you know the functions of these particular listed organelles. So let's go back to our typical illustration of a eukaryotic cell. Now, looking at our illustration, we can see that the nucleus is front and center. And as we previously discussed, in there you will find all your DNA, all your genomic information. You could say your chromosomes, your chromatin, however you refer to it. That is where you're going to find your genetic code. The nucleus is then able to dictate what is happening with the other organelles that we can find floating in the cytosol of the cytoplasm. And what I want to take a few minutes now is discuss what each organelle contributes to the story. So let's start with our control center, the nucleus. Since that is where we find our DNA, that will be the starting point of getting directions of anything that needs to be produced or developed within the cell. So let's say, for instance, this morning when you woke up, one thing that your body did is it's figured out what hormones or enzymes are required. And let's say, for instance, we are interested in looking at a cell and how that cell is able to produce testosterone or estrogen. Well, the first step will always be to go to the nucleus and take a look at the DNA to find the proper directions. Now, not all your DNA is useful, but what we're looking for are the quote unquote genes, and the genes will be sections of DNA that are able to do transcription and translation, which means that there are sections of DNA that we can utilize for certain entities within the cell. Once the proper strand or location of DNA is found, we now see that the DNA cannot leave the nucleus, so we're going to go ahead and transcribe the DNA into RNA. RNA, which we'll think of it as a copy of our directions, is able to go in and out of the nucleus through the use of the nuclear pores found on the nuclear envelope. So here you can see the little cavities. So please keep that in mind. DNA cannot leave the nucleus, and that is why it will make RNA, and RNA will be a copy of the directions which can go in and out of the nucleus as it pleases. You may also notice that inside of the nucleus there's a smaller blue circle. That is the nucleolus, and the nucleolus is where your ribosomes are going to be assembled. The ribosomes are going to play an important role in our protein production. All right, now back to our nucleus. So we have our directions. We went from DNA to RNA, and the RNA is now going to leave the nucleus, and it's going to start interacting with the other organelles in our cytoplasm, starting off with our ER or endoplasmic reticulum. We have two versions of endoplasmic reticulum. We have rough and smooth. Rough because when we look closely at the structure, we see that it's studded with ribosomes, these little orange dots everywhere. And that is because the rough ER is able to produce proteins that the cells might need. So rough ER is studded with ribosomes, and it's going to help us make any proteins that the RNA is coming down saying it needs for its secretion or function. The smooth ER does not have any ribosomes on the surface, and that's because its primary function is to produce lipids. It also helps with detoxifying enzymes, but for the sake of our conversation, this is where we're going to go for any lipids that are required. Once the protein and the lipids are collected, then the next step will be to kind of combine them and separate them and join them in their final secretion. In order to do that, we're going to have to visit this green structure right here. This is the Golgi apparatus. Some of you might know it as the Golgi complex, which is perfectly fine. I want you to think of this kind of like the UPS or the post office store. This is where all the individual pieces, the proteins, the lipids get together. They get sorted, they get aligned, and they get organized into the final secretion. In fact, if you take a look at the shape of the Golgi apparatus, you'll see that it's flattened sacs, and out of them are coming little vesicles, little containers that are filled with whatever final secretion the cell is trying to produce. And in this case, the little vesicles are making their way over to the cell membrane, where they are then released, or they're doing exocytosis. And you can see the excretions are released. So this would be, for instance, our hormones that are directly released into the bloodstream. Or if we're looking at milk production, it's released into the milk ducts, or any type of enzymes or whatnot that are required. So that is the sequence of events that we always see. Whenever a secretion needs to be produced, we're going to go ahead and start off with the nucleus because that is where we're going to find our DNA. DNA will make a copy of itself in the form of RNA, and from there on, we'll go shopping. We'll go shopping for our individual ingredients. The rough ER will give us proteins, the smooth ER will give us lipids, and they are all combined together into the Golgi apparatus, which will sort, package, and send the final product on its way. And in case you're wondering, yes, sometimes the final product does stay within the cell itself. Now, there are some other things floating around, primarily here in orange. We have our mitochondria. The mitochondria is the powerhouse of the cell. It's going to be able to produce lots and lots of ATP molecules for us, which allow us to provide different versions of chemical energy. We also have things like our lysosomes. Lysosomes are more known as kind of like a little housekeeping gene. They are able to hydrolyze any type of debris or a damaged organelle that you might find in the cell. I don't see a picture of it, but it's going to be mentioned in our chart coming up. We also have peroxisomes. Peroxisomes tend to produce hydrogen peroxide as a method to do a cleanup, and they are really good at addressing blood toxins. So you tend to find a lot of them in your kidneys and in your livers. I also want to mention, and you know what, I'm going to have to erase some of my little markings here. I want to mention the centrioles that I see tucked away. Here we go. So these protein complexes are going to be very important for allowing for scaffolding and moving of the organelles, as well as in a little bit, we're going to talk about DNA replication. And when that happens, we're going to need a mechanism to kind of sort through the chromosomes and separate them for the upcoming daughter cells. That's where the centrioles are going to play a very important role in moving the DNA from side to side. Now, on the side of my PowerPoint, I see I made a mention of the DNA and product development, so we've talked about that already. Let's tackle the endosymbiotic theory. And in order to do that, I think I'm actually going to go over to the chart. Here is the table that outlines many of the organelles that we've kind of chit-chatted about, so right? So here you can see your Golgi apparatus. Here is your rough and your smooth ER. And within the functions that we can talk about, just a reminder, rough ER is going to be for protein production. Smooth ER is going to be for your lipids as well as for purification or detoxification, I could say. Golgi apparatus is going to be our UPS store, so it packs everything and produces our final product. Here are the ribosomes that are being produced by the nucleolus. And remember, they play a very important role in protein synthesis. And then, oh, here we go. Here are our peroxisomes. So these are the ones I was mentioning. We'll go ahead and secrete hydrogen peroxide. And they're very important for blood toxins to get rid of it out of the cell. On top, we have our mitochondria, and that, of course, is used for ATP synthesis. We call that the powerhouse of the cell. And the reason that I wanted to show you this is because when you look at the illustration of the mitochondria, I want you to look at a unique feature, which is that it has a double membrane. So when you look upon closer, you see that the inner membrane is the one that has all the curvations, and then the outer membrane is a smooth covering. Now, that's unique because many organelles have just one membrane, but the mitochondria has a double membrane. It also has another unique feature, which is that mitochondria has its own DNA. It's termed mitochondrial DNA, and it is exclusively inherited from your mother. So the nuclear DNA, the DNA that you have in your nucleus, is part mom, part dad. But the mitochondrial DNA is only from mom. So it's maternal DNA. Because of the fact that mitochondria has its own DNA, meaning that technically it can function by itself and not have to rely on directions from the nucleus, and it has a double membrane, scientists were very intrigued to figure out why these abnormalities were there. And based on research, they were able to establish the endosymbiosis theory. The endosymbiosis theory says that millions of years ago, certain organelles were free living cells. These cells were engulfed by a larger cell, and the larger cell then made it part of its genome. And when I say that, I always visualize like Pac-Man, and I visualize the Pac-Man just eating the little dots as it's moving along and you're playing the game. And that's exactly what the host cell did. It engulfed the organelle, or I should say it engulfed the smaller cell and made it part of its genome. Now, how do we know this happened? Well, we know this happened because of the two pieces of evidence that we were talking about previously. The organelle, aka the mitochondria, has its own DNA, And the double membrane indicates that the inner membrane was part of the original structure. The outer membrane, which is nice and smooth, was placed around by the host cell to indicate that it now was part of its genome. And fun fact, the outer membrane of the mitochondria doesn't do anything. It's literally just a structural entity. All the work is done by the inner mitochondrial membrane. There is another organelle that's used for the endosymbiosis. We are not owners of those, and that would be chloroplast. Chloroplast, which you find in plants and other autotrophs, that organelle also has its own DNA and also has a double membrane structure. So yet another example of the endosymbiosis theory. Continuing on in our table, we can see we have our lysosomes that were mentioned for the production or the hydrolysis process to get rid of any type of debris or no longer functioning organelles. We see our cytoskeleton, which I did not mention in the image before, but here is the microtubules, intermediate filaments, and microfilaments. These are all protein structures that are important for the overall shape and structure of the cell. They can also be used for the formation of like cilia or flagella, and also for scaffolding to allow for intracellular movement. Here are the centrioles, which I highlighted, and these, of course, are going to be important for forming a network upon which we can move our organelles. We can also utilize it for the movement of the DNA in a little bit when we talk about DNA replication. And in the final part of our table, we see some of the cellular extensions. So here is cilia, flagella, and microphyllae. These are excellent for protecting open surfaces, providing movement in the case of the flagella. And microphyllae is really great for allowing absorption to occur by increasing the overall surface area that the items have access to. And then finishing it off, we can see we have our nucleus again, and it just highlights the fact that it is surrounded by a nuclear envelope that has little pores in it. Remember that DNA cannot leave the nucleus. That is why we are making a copy of it, and then RNA is able to go in and out. And then, of course, that the nucleolus is going to be our site for ribosome production. I also want to keep in mind or remind you real quick that not all your DNA is able to do the RNA copying, which is referred to as transcription, which we'll cover in the third part of our lecture. And that part is the part of the DNA that's able to do that is referred to as genomic DNA because that's where we have our genes. The rest of the DNA is actually referred to as non-genomic DNA. They used to refer to it as junk DNA. We now know that it's definitely not junk because it's there to stabilize the genomic DNA and assist with its replication. And a lot of scientists also use that DNA to indicate proof of evolutionary changes. In case you are interested, what percentage of your DNA is genomic versus junk? Many scientists agree that genomic DNA is around 2% of your DNA. Yeah, not a lot of it, but look how much we can do with just that little 2%, right? And our last illustration is termed chromatin. Chromatin is coiled up DNA. We have a massive amount of DNA in each of our cells, so the cells have to come up with an effective way to kind of store it. And in order to do that, there are different ways to condense the DNA down. Chromatin is just one of them. And in fact, I think we can see the story if we go over to our next slide. And what we're seeing here is a version of the chromatin that continues to coil up to eventually give us the typical chromosome shape that we are so used to. Now, how do we get to this point? Well, I'm going to show you that on the next illustration. And in order to do that, we want to keep these key terms in mind, okay? So we want to be able to explain what is a histone, what is a nucleosome, what is chromatin, and what is chromatids. All right, let's go take a look. So here is what we call naked DNA, which is the typical double helical structure that we're so used to seeing. We have the sugar phosphate backbone, and then we have the little nucleotides that are connecting it, A with T and G with C. Remember your law of complementary base pairing, which says that in order for DNA to be the most stable, the adenine will have to bind with thymine and the guanine will have to bind with cytosine. Can you make other matches? Definitely, but that will cause instability in your DNA molecule. So the first part is to pair up the nucleotides. And now that you have your beautiful strand of DNA, on average, each cell will have over 6 feet of DNA inside of it, massive amount. So we're going to have to coil it down and condense it as much as we can. So the first thing that we see happening is that the naked DNA is going to go ahead and start wrapping itself around histones. Histones are storage proteins, and they will go ahead and will utilize it in sets of 8. You can see them stacked up here, 2 sets of 4. Once the DNA wraps itself around the histone, we call that a nucleosome. So a nucleosome is a strand of naked DNA wrapped around histones. And these histones will then start to stack on top of each other, and they will create chromatin. So chromatin is coiled up DNA in the form of nucleosomes. And it really starts to look like an old school telephone cord where the fibers will just coil on top of each other and the chromatin will just continue to condense and condense and condense. So here at the bottom of our illustration, we see that we have the chromatin, the long strand of DNA that is being shuffled down or condensed. And this is how your DNA will stay within your nucleus. Now, the other thing that I want to tell you is eventually you will see that a lot of cells will need to replicate their DNA because they want to obviously continue on and do things like mitosis and meiosis. When they replicate their DNA, we see that the chromatin will hold on to the copy created, so the double DNA, through its centromeres. So here when we look at the illustration, we see that we have 2 chromatins that are being held together by the centromere. And these are duplicate versions of each other. So we can then go ahead and say that these are sister chromatids. So chromatin, Tin, is when you have your coiled up DNA. And chromatids indicates that you have, quote unquote, a duplicate version that are attached to each other. And that is why they're called sister chromatids. And that will then sit and or be done in efforts for the cell to eventually continue on and complete mitosis or meiosis and create a new line of cells or some more what they call daughter cells. So go ahead and take a look at the illustration, and once again you can see your naked DNA on top, and as it coils it will wrap itself around histones making nucleosomes, and then the nucleosomes are condensed further into chromatin. And from chromatin, if we make a copy of it, we then have chromatids. So sister chromatids are duplicated chromatins that are being held together by the centromere. Now that we know all this, let's continue our journey on part 3, where we're going to take a look at DNA replication, and we're also going to talk about the cell cycle as well as transcription and translation. I will talk to you soon, and as always, please do not hesitate to reach out if you have any questions, comments, or concerns.